Welcome to EPG Pathshala. I am Aparna, working as scientist in MFPI Food Quality Control Laboratory, Professor Jaishankar, Telangana State Agricultural University, Hyderabad. Today, we will learn about food preservation using bacteriocins. Ever since the era of Louis Pasteur and Robert Koch, there has been scientific recognition of an essential need to control the detrimental microorganisms which are present in our environment. And the discovery of penicillin by Alexander Fleming in 1929 opened the door for usage of therapeutic antibiotics by the medical and veterinary communities to combat specific disease-causing microorganisms. Although therapeutic antibiotics are prohibited for use in the foods, the utilization of antagonistic additives with preservative or antimicrobial properties has become a landmark approach in food safety and preservation. In food beverages, addition of antimicrobial compounds to processed products has become a traditional weapon in the food preservation arsenal. Comprising a subgroup within the far larger body of commercial food preservatives are the bacteriocins. Bacteriocins are produced by bacteria and possesses antibiotic properties, but bacteriocins are normally not termed antibiotics in order to avoid confusion and concern with therapeutic antibiotics that can potentially elicit allergic reactions in the humans. Bacteriocins, though are antibiotics, are exclusively used only in food processing and food industry applications. Bacteriocins differ from most therapeutic antibiotics in being proteinaceous and generally possessing a narrow specificity of action against the strains of the same or closely related products. Bacteriocins are ribosomally synthesized polypeptides possessing bactericidal activity that are rapidly digested by proteases in the human digestive tract. Bacteriocins are a heterogeneous group characteristically selected for evaluation and use as specific antagonists against problematic bacteria. However, their effectiveness in food can become limited for various reasons, and the cost remains an issue impeding broader use of bacteriocins as food additives. Let us now see the ecology of bacteriocins. On an evolutionary basis, it appears that the ability to synthesize one or more bacteriocins has been a highly advantageous characteristic. A clear opportunity for survival and proliferation of an organism can be envisioned if it can eliminate a competing organism given the diversity of species and rapid growth of bacteria. Low molecular weight antibiotics like tetracyclines, lytic agents, toxins, bacteriolytic enzymes, bacteriophages, and metabolic byproducts such as organic acids, hydrogen peroxide, diacetyl also function in somewhat similar capacity, but nonetheless the capability to produce bacteriocins and producer cell immunity occurs abundantly only in the prokaryotes, both eubacteria and also the archaebacteria. Bacteriocins play a fundamental role in bacterial population dynamics, even though the degree of bacteriocin interactions is also complex at the ecological and evolutionary levels in mixed populations. Now let us try to classify the bacteriocins. We will go ahead and understand the classification of bacteriocins. First discovered by Gracia in 1925, Principi V was produced by I strain of Escherichia coli against another culture of E. coli. The term colicine was coined by Gracia in 1946 and bacteriocin was used by Jacob and others in 1953 as a general term for highly specific antibacterial proteins. The term colicine implies a bactericidal protein produced by varieties of E. coli and closely related Enterobacteriaceae. Bacteriocins were originally defined as bactericidal proteins characterized by lethal biosynthesis with a very narrow range of activity and absorption to specific cell envelope receptors. Later, the recognized association of bacteriocin biosynthesis with plasmids was added to the description. The definition has since been modified to incorporate the properties of bacteriocins which are produced by gram-positive bacteria. Bacteriocins from gram-positive bacteria commonly do not possess a specific receptor for adsorption, although exception exists and are more frequently of low molecular weight than the colicins have. Today, the bactericidal peptides or proteins produced by the bacteria are typically referred to as bacteriocins. Usually, to demonstrate the proteinaceous nature of a newly characterized bacteriocins, sensitivity to proteolytic enzymes such as trypsin, chymotrypsin and pepsin is an accepted demonstration. Evaluation for use as a food additive requires estimation of its heat resistance given the widespread use of thermal processing in food production. Over the years, there are lots of research publications who have reviewed the colicins, the bacteriocins from LAB and applications of bacteriocins. Most of the bacteriocins from LAB, lactic acid bacillus, are cationic, hydrophobic or amphiphilic molecules which are composed of 20 to 60 amino acid residues. And these bacteriocins are commonly classified into three groups which also include bacteriocins from other gram-positive bacteria. So the examples of the bacteriocins are categorized into three classes as you can see from the table 1. The first group is the lantibiotics from the lanthionine containing antibiotic, and these are small peptides containing unusual amino acids lanthionine and also methylanthionine and dehydrolenine and dehydrobutyrene. These bacteriocins are grouped in class 1. Class 1 is further subdivided into type A and type B. The type A antibiotics are elongated peptides with a net positive charge that exert their activity through the formation of pores in bacterial membranes. Then you have the type B antibiotics, which are smaller globular peptides and have a negative or no net charge antimicrobial activity, which is related to the inhibition of specific enzymes. After antibiotics, you have the second category, that is the small heat stable non-lanthionine containing peptides, and they are called as class 2 bacteriocins. And the largest group of bacteriocins in this classification system have got peptides which are further divided into three subgroups. The class 2A pediocin-like peptides have an N-terminal consensus sequence, and this subgroup has attracted much of the attention due to their anti-listeria activity. Class 2B contains bacteriocins which require two different peptides for the activity. Then you have the class 2C containing the remaining peptides of the class, including the dependent secreted bacteriocins. Then you have the class 3 bacteriocins which are not very well categorized, and this group houses larger heat stable proteins that are of less interest to the food scientists. So for food applications, it is only the class 1 and class 2 bacteriocins which are used for food processing, preservation and other kinds of applications. You also have a fourth class consisting of complex bacteriocins which require carbohydrate or lipid moieties for activity and has also been suggested by some scientists. However, bacteriocins in this class have not been characterized adequately at the biochemical level to the extent that the definition of this class requires, and additional descriptive information is not available about the class 4 bacteriocins. So after learning about the classification of bacteriocins, let us now move ahead for the next topic. Let us now try to understand the mode of action of the bacteriocins in food. The antibiotic activity of bacteriocin from gram-positive bacteria is based on interaction with bacterial membrane. Some of the bacteriocins elaborate amphiphilic property, generalized membrane disruption by the pore formation. Lactic acid bacteria produces several types of pore-forming peptides. Most of the bacteriocins produced from lactic acid bacteria are bactericidal peptides which act primarily by creating pores in the membrane of their target cells. Although the formation of the pore is a general feature, the size, stability and conductivity of these pores differ considerably from bacteriocin to bacteriocin. The formation of poration complexes causes an ionic imbalance and leakage of the inorganic phosphate. These mechanisms rely upon stabilizing interactions between the membrane phospholipids and the cationic residues of the peptides, allowing the insertion of hydrophobic regions into the outer leaf of the membrane. One associated with the membrane surface of a number of ordered bacteriocin could potentially aggregate. The bacteriocin complex can in principle completely span the membrane, thereby forming a transient pore in which there is dissipation of proton motive force, which involves the partial or total dissipation of either or both the transmembrane potential in the target organism. Anyway, most bacteriocins interacts with anionic lipids that are abundantly present in the membrane of gram-positive bacteria. And these anionic lipids may enhance the conductivity and stability of antibiotic pores as a docking molecule or may act as receptor in the class II bacteriocins. Now let us try to understand the medical significance of these bacteriocins. Bacteriocins are of interest in medicine because they are made by non-pathogenic bacteria that normally colonize the human body. Loss of these harmless bacteria following antibiotics use may allow opportunistic pathogen bacteria to invade the human body. Bacteriocins have also been suggested in cancer treatments, and they have shown distinct promise as a diagnostic agent for some kinds of cancers. Bacteriocins were also tested as AIDS drugs around 1990 but did not progress beyond the in vitro tests on the cell lines. As with today's antibiotics, bacteria can evolve to resist the bacteriocins. However, they can be bioengineered to regain their effectiveness. Further, they could be produced in the body by intentionally introducing the beneficial bacteria such as probiotics. Let us now try to understand how the production of bacteriocins can take place. There are many ways to demonstrate the bacteriocin production depending on the sensitivity and labor intensiveness which is desired. To demonstrate their production, technicians inoculate multiple strains on the separate multiple nutrient agar Petri plates and incubate them at 30 degrees centigrade for 24 hours, overlay each plate with one of the strains and incubate again at 30 degrees centigrade for 24 hours. After this process, the presence of bacteriocins can be inferred if there are zones of growth inhibiting around the stabs. And this is the simplest and least sensitive way. It will often mistake phage for bacteriocins. Some methods prompt the production with UV radiation, mitomycin C or heat shock. UV radiation or mitomycin C are used because the DNA damage they produce stimulates the SOS response. And cross-steaking may also be substituted for lawns. Similarly, production in broth may be followed by dripping the broth on a nascent bacterial lawn or even filtering it. Then now let us try to understand the application of bacteriocins in food preservation and other food applications. The principal physical, chemical, enzymatic and microbiological reactions which are responsible for food deterioration are very well known. Various preservation techniques to avoid different forms of spoilage and food poisoning, including reduction in the temperature, water activity and pH, as well as addition of preservatives such as antimycotic, inorganic or organic compounds, are all known to slow or prevent the growth of microorganisms in any kind of food matrix. Nisin is one bacteriocin which is produced by Lactococcus lactis subspecies. Lactis has been applied as a food preservative in several countries. It has been used to control some kinds of foodborne pathogens, especially some species of genera like Aeromonas, Bacillus, Clostridium, Enterococcus, Listeria and Staphylococcus. The potential application of bacteriocins is consumer friendly. Biopreservatives, either the form of protective culture or as additives, is significant besides being less potentially toxic or carcinogenic than the current antimicrobial agents. Lactic acid bacteria and their byproducts have been shown to be more effective and flexible in several food applications. In addition of the functional properties in the lactic acid bacteria, they also improve the preservative effect and also add flavor and taste to the food in which they are added. Consumers have been consistently concerned about possible health effects from the presence of chemical additives in their foods. As a result, consumers are drawn to natural and fresh foods with no chemical preservatives which are added. This perception, coupled with the increasing demand for minimally processed foods with long shelf life and convenience, has stimulated research interest in finding natural but effective preservatives. Bacteriocins, which are produced by lactic acid bacillus, may be considered natural preservatives or biopreservatives that fulfill these requirements. Biopreservation refers to the use of antagonistic microorganisms or their metabolic products to inhibit or destroy the undesired microorganisms in the foods to enhance safety and extend the shelf life of the food products. Three approaches are commonly used in the application of bacteriocins for biopreservation of the foods. The first one is inoculation of food with LAB that produce the bacteriocins in the products. The ability of the LAB to grow and produce the bacteriocin in the products is crucial for its successful use. Then the second approach is addition of purified or semi-purified bacteriocins directly as food preservatives. Then the third application is use of a product which is previously fermented with a bacteriocin producing strain and adding it as an ingredient in the food processing. Let us now learn about the hurdle technology to enhance food safety, where bacteriocins also are added as one of the hurdle to enhance the food safety and also preservation. The major functional limitations for the application of bacteriocins in foods are their relatively narrow activity spectra and moderate antibacterial effects. Moreover, they are generally not active against gram-negative bacteria. To overcome these limitations, more and more researchers use the concept of hurdle technology to improve the shelf life and enhance the food safety. As you can see from the table 3, bacteriocins like nisin are used in combination with either heat treatment, antimicrobial agents, antioxidant agents, chelating agents to get the desired effect along with the combination of a bacteriocin. It is well documented that gram-negative bacteria become sensitive to bacteriocins if the permeability barrier properties of their outer membranes are impaired. For example, chelating agents such as EDTA can bind the magnesium ions from the lipopolysaccharide layer and disrupt the outer membrane of gram-negative bacteria, thus allowing the nisin to gain access to the cytoplasmic membrane. Similarly, you can see the other kinds of hurdle concepts in the table 3. Now let us learn about the bacteriocins and their applications in the packaging films. Incorporation of bacteriocins into packaging films to control the food spoilage and pathogenic organisms has been an area of active research. Antimicrobial packaging films prevent the microbial growth on the food surface by direct contact of the package with the surface of the foods such as meats and cheeses. For this reason, for it to work, the antimicrobial packaging film must contact the surface of the food so that the bacteriocins can diffuse to the surface and create the preservative effect. The gradual release of bacteriocins from a packaging film to the food surface may have an advantage over dipping and spraying foods with the bacteriocins. In the later processes, antimicrobial activity may be lost or reduced due to the inactivation of bacteriocins by the food components or dilution below the active concentration due to migration of the bacteriocins into the food matrix. Two methods have been commonly used to prepare the packaging films with the bacteriocins. One method is to incorporate the bacteriocins directly into the polymers. Examples include incorporation of nisin into biodegradable protein films. Then you have the second category, that is the packaging film forming methods. Here, heat press and casting were used to incorporate nisin into films made from the soya protein and corn zein, and they were tested. Both the cast and heat press films formed excellent films and inhibited the growth of Listeria plantarum and compared to the heat press films. Compared to heat press films, the cast films exhibited larger inhibitory zones when the same levels of nisin were incorporated. And incorporation of EDTA into the packaging films also increased the inhibitory effect of nisin against Escherichia coli. So today we have learnt about what are bacteriocins, what are their effects as preservation, the mechanism of action of bacteriocins, and we have also seen the hurdle concepts by using the bacteriocins. So to sum up the entire topic, preservation by bacteriocins, as you all know by now that bacteriocins represent one of the best studied microbial defense systems. Although we are still in the earlier stages of exploring their evolutionary relationships and ecological roles, it is clear from their abundance and diversity that they are microbial weapons of choice for food processing, preservation, and various applications in the food industry. Sorting out why there are such a successful family of toxins will require a substantial commitment to the future research and applications of bacteriocins on such kind of toxins. It is expected that a better and deeper understanding of the molecular basis of the antimicrobial activity of bacteriocins will definitely result in safer food in the near future by usage and application of bacteriocins in preservation as well as in packaging. Following a knowledge-based approach, a new biopreservation strategies as well as unique biotechnological applications of these natural antibiotics are envisaged into a new and further applications in the food industry. For further information on bacteriocins, you can refer to exclusive books on food preservation or bacteriocins and applications in food industry. Thank you.